Hello, in this vodcast we're looking at genetic manipulation. This is the second of two vodcasts on genetic engineering. And our objective for this vodcast is that we will understand how a gene can move from one individual or species into another and why this is done. There are two kinds of genetic engineering. The one that we covered in the previous vodcast is called artificial selection or selective breeding or just breeding. And for this one, the desired trait is acquired by breeding chosen individuals. So we're working with thousands of genes at the same time since we're just working with specific individuals, and this type of genetic engineering has been going on for all of history and probably beyond history. The second kind is called genetic manipulation, and this is when people generally refer to genetic engineering, this is what they're talking about. And for this, it's engineering for a particular gene, where basically a gene is located for a desired trait, and then that gene is taken out of one individual and is placed in different individuals or a whole new species. Cloning is one of the methods used in genetic manipulation. It's the production of genetically identical copies of a cell or an individual. One other term that you need to know is transgenic organism, and a transgenic organism is an organism that contains genes from another species. Restriction enzymes are enzymes that basically permit us to do genetic manipulation. These enzymes evolved in bacteria, and they use them as their form of immune system, and they basically just cut up DNA. So if a virus injects its DNA into the bacterium's restriction enzymes will restrict that type of DNA by cutting it up into little pieces, and when it gets cut up into little pieces, that will kill the virus and will stop the viral DNA from taking over the bacteria. And scientists use these restriction enzymes. What they do is if you look at this diagram over here, there's several different restriction enzymes. Each one's active site will look for a specific active site. Echo R1 looks for the sequence CTTAAG, and what it does is whenever it finds that sequence, it cuts it. And it cuts it. Now, some restriction enzymes will cut it straight down. What Echo R1 does is that you see how it doesn't cut in the same place on the different strands? What that leaves are sticky ends, and these are very useful in genetic engineering because those sticky ends can then stick to. It's a place where they can more easily stick to other DNA which have been cut by the same restriction enzyme. So these restriction enzymes are used to prepare DNA for electrophoresis. In the State Lab Biodiversity, we have simulated this. And also, as I said, that it's used to cut DNA for genetic manipulation. Now here's a summary of how genes are manipulated. One vocab word that you need to know is the word plasmid, and a plasmid is a small circular chromosome of DNA, very small, and it's found in bacteria. Bacteria have one large chromosome, and they will have several small plasmids. Now what happens is, first thing that's done is that the desired gene and the plasmid are cut with the same restriction enzyme. It has to be the same restriction enzyme, or else when the two are mixed together, then they won't be able to join together. If they're cut with the same restriction enzyme, then they will be able to join together. And you'll end up with a recombinant plasmid once the enzyme called ligase glues them together. And it's called a recombinant plasmid because the plasmid's been recombined with foreign DNA. Then the plasmid is put into a vector, which would be a bacterium or a virus, and then the gene is then cloned by the bacteria itself, making multiple copies of the plasmid, or also by the bacteria itself reproducing, and that way also making multiple copies of the plasmid. Now I'm going to show you a video that summarizes how genetic manipulation occurs.
Using genetic engineering, scientists can create cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
Which brings us to cloning, because that was one part of cloning. First is natural cloning. Natural cloning is basically asexual reproduction, and like all the types we talked about before when doing the reproduction unit, and also identical twins are also a method of cloning. Then there's gene cloning, and gene cloning is where you're making copies of the genes, and that's what we just discussed. Reproductive cloning is where a copy of an entire animal is made. And the last kind of cloning is called therapeutic cloning, and for this, embryonic stem cells are then used to create tissues that can replace diseased or injured tissues. This area right now is very, very controversial, because the way you get embryonic stem cells is by taking it from an embryo. The way you take it from an embryo is you basically have to kill the embryo, and people who consider an embryo to be fully human life consider making embryonic stem cells to be murder. And other people say, well, that even if you consider them to be fully human, you're saving thousands and thousands of lives as opposed to not killing an embryo, which would never come to term anyway. Now, here are some uses of genetic manipulation. One would be organ or tissue transplants. There are some organs or tissues where people need to wait for years and years and possibly never get before they die of a particular disease. And if you could, say, through genetic manipulation, make a pig that doesn't have the things which would make us reject their organs, pig organs could then be used for transplants. Or also, you could take, say, one person needed a new kidney, then all you have to do is just take embryonic stem cell and grow a whole new pancreas and put that in. Or you could take your own cell. You could take your own cell, say a skin cell, and put that into an embryonic stem cell. And then when you grow a whole new kidney, that kidney is genetically identical to you because it comes from your own nucleus, and it won't be rejected. You won't have to take any immunosuppressing drugs like people who have organ transplants today now have to do. Crop improvement is another major way that we use genetic manipulation, and unlike organ transplants, this is something that's not in the future. It's something that is in wide use today. Rather than spraying an entire field with pesticides, then you just take the gene that produces the pesticide and genetically manipulate that into the crop plant, and now you don't have to spray and kill many insects that are very beneficial insects. Instead, the plant has the pesticide as a part of it, so that pesticide will only kill those insects that try to eat that plant. Also, genetic manipulation can be put into vaccines, where For example, you could put the vaccine for something into a completely harmless bacterium, and that completely harmless bacterium will then produce the antigen, and you'll become immune to that disease. Hepatitis B is an example of that kind of vaccine. So instead of using a weakened pathogen, you're not putting a pathogen in at all. Another way you can get the vaccines instead, you could have, say, a potato produce the antigen, and then when your children go to the doctor to get their vaccine, instead of getting a shot, they'll eat some potato chips. Here I'll show you a video right now on some of the use.
1995, Ryan Cranny and farmers like him welcomed the news of an agricultural breakthrough that promised to cut down their use of sprays. Monsanto, the world's biggest biotechnology company, came up with a much less toxic method for killing one of the potato's most deadly enemies, the Colorado potato beetle, which can pick the leaves off a plant virtually overnight.
Colorado potato beetle worldwide is probably the most serious insect pest in potatoes. We still estimate that, out in the western U.S. and probably across the U.S. as a whole, about 40% of the insecticides that were applied for Colorado potato beetle control.
Monsanto's innovation was to create a new kind of potato called the new leaf potato. It was the first potato to be genetically engineered to contain genes from a different biological species.
Genetic engineering is a radically new technology compared to traditional breeding. It allows us to move genes without regard to species barriers. It allows us to move a gene from a butterfly, into a corn plant, from a starfish into a wheat plant.
Monsanto's new leaf potato used a gene from a common soil bacterium, one that makes a protein that kills potato beetles without causing harm to humans. The bacterium is called Bacillus thuringiensis, or BT for short. To help market its BT potato, Monsanto hired plant physiologist Michael Thornton to be one of its liaisons to farmers in Idaho.
Monsanto was able to identify the gene in that bacterium, the BT gene that was responsible for production of that protein. And they could use a process to insert that gene into a potato variety, one that growers were already familiar with. The beetle eats that leaf and gets that BT protein inside it, and it disrupts its digestive system. So then that eventually kills the Colorado potato beetle.
The promise here was that you could diminish spraying. You might pay a little bit more for these potatoes, but since they generated their own pesticide, you could give up some of your spraying. And this was very attractive to a lot of potato farmers.
Now we come to our concluding questions. Number 1, state one difference between selective breeding and genetic manipulation. Number 2, what is the function of a restriction enzyme? Number 3, what is a recombinant plasmid? Number 4, what kind of cloning is used in genetic manipulation? Number 5, why do farmers prefer to use genetically engineered or another way of putting it is genetically manipulated crops?